With this experiment, we wanted to investigate how these severe droughts, which are increasing worldwide, do affect a tropical ecosystem. The impact of these severe droughts on our forest is very important because forests sequester a large amount of carbon on our planet, and just the tropical forests are the lungs of our planet, and their function is very important. Today we don't really know a lot about how these extreme drought events impact forest functioning, and that was the aim of this big experiment, which was conducted with a series of researchers worldwide and which I coordinated with my team leaders Nemaya Lett and Laura Meredith from the Biosphere 2. The aim of this study was to really detect the different fluxes and pools throughout the drought period. So we implemented a huge measuring network within the Biosphere 2 tropical rainforest, measuring everything from the leaf fluxes to the trunk fluxes down to the soil and root, and connected this to isotope lasers so we could also measure the isotopic signature of all these different carbon and water fluxes, as well as volatile organic compounds, so small molecules which are emitted into the atmosphere, and we could trace these throughout the drought. As the really innovative part of this experiment, we added a tracer of stable isotopes. They are non-radioactive, so we did a pulse labeling experiment of 13CO2 to see how carbon would travel, being uptaken from the leaves, travels through the trunk, being respired by the root and by the soil microbia. And in the end of the drought, we also wanted to investigate how the system responded to a re-wetting, and especially how the deep water reserves were used. So we added another water tracer to the bottom of the forest. We also monitored the volatile organic compound dynamics in this system. Volatile organic compounds are small molecules which can be emitted by plants or even by soils or uptaken by soils, which can protect plants against severe drought. For example, heat stress and light stress. And what was really interesting is that we found an enormous dynamics throughout the drought period. Actually, different VOCs peaked during different times. We had a cascade from isoprene to monoterpene to hexanol, indicating this increase in stress severeness. On the other hand, the 13C pulse labeling showed us that even under drought, when photosynthetic carbon uptake is strongly reduced, the production of these VOCs is maintained at a high level from this fresh photosynthesis, showing us that the importance of producing these molecules as stress protection is very high. Similarly, we saw that the soil responded to these changes in atmospheric VOC conditions. The soil was a strong sink of VOCs, so soil microbes obviously used these VOCs as carbon sources, but shifted their carbon source towards the changes of different VOCs throughout the drought periods. So again, this is an important marker to understand how drought impacts our system, and it has an effect on atmospheric chemistry. So there is a huge interaction between the ecosystem response and atmospheric processes further driving climate change. What were our main results? What was really interesting is to understand the diversity of responses within this tropical rainforest. Overall, we could categorize the different tree species into 4 response types. So some were very drought sensitive, they were very active in the pre-drought period, but showed severe signs of drought stress impact early on when we dried down the system. So we had leaf shedding, it was like here in autumn the leaves were dropping. Other trees were much more resilient, and actually they had a more constant reaction, which then took a more important proportion throughout the drought. Overall, we could also see that the understory species were actually covered from the severe drought impact because they were in the shaded, more humid environment. So also the positioning within the forest strata has an important function on their responses. Regarding the re-wet, it was extremely interesting that those trees which suffered a lot in the early drought when the upper soil layers dried out did actually have access to the deep water reserves. It shows that the system actually spares these deep water reserves until late to drought, and only under really severe condition these deep soil moisture layers are used. The other interesting aspect was that it took pretty long during the re-wet phases until this very drought-stressed system took up the deep water reserves and transported them to the leaves, and it took 4 to 6 weeks in some plants before this water was actually respired. So we also found relatively long legacy effects, especially in the very active plants, which took more than 3 months to recover back to their pre-drought values. The understanding of the different adaptation of plant responses in forests is extremely important to understand forest functioning worldwide. I think in each forest we have different response types, so it's not just the biodiversity of different plant species, it's really the diversity in plant functions which we need to understand to see what are the mechanisms which buffer our systems against severe drought, and what may even accelerate, what are the interactions within the plant, soil, atmosphere system which drives the system into their limits or also buffers them from extreme drought impact. So this knowledge is quite important for further research to better characterize these functional response types within our forest.